Launched. And get my mouse to stop being caught up. There we go. Alrighty. Get that open. All right, all the things that need to be open are open. All right, some of you have submitted some late assignments. I will get to those and get you caught up. And again, we are on the last couple of weeks. So if you have anything outstanding, please make sure you don't wait to the last minute to get those things caught up. All right, so again, we are in the new module this week called DNA analysis. Most of y'all have taken biology already, so you probably familiar with DNA as the macromolecule. It is the structure in our bodies, in our cells, that has all of the genetic information. So it tells us, it's like the blueprints on you or on living things, and it tells it how to make it and what everything should look like in the organism. So it's like the blueprints of the body. Somebody wants to see how a building is made, they pull up the blueprints and look at how it was constructed. DNA is like that for you. Excuse me. So from DNA, they can use it to analyze exactly who the person might be related to. And this is new science. So a lot of cold cases are being solved because all of a sudden now that DNA technology is available, they have evidence that they've been saving for a long time. They can now take that evidence and hydrate it and check for the DNA. There is something called the human genome, which is like a big data bank of all the different arrangements of what your genes can look like. So they know what the genetic structure is for each kind of gene. And they can, when they do the DNA analysis of the individual, they can compare it to the information in the genome. So like I said, this is like a huge data bank. Somebody has gone in and spent years trying to figure out what each gene looks like genetically. Okay. All right, so the first unit is going to, like I said, there's a lot of vocabulary in here. The first unit gives you a little biological background on what DNA is. And like I said, this is something that you might have already remembered from biology. So this is a quick review. So take a look at that. Because like I said, this is basically biology background information. Okay. So we know that the DNA is found on the chromosomes, and the chromosomes are found in the nucleus of the cell, and the chromosomes will duplicate and create new individuals from the information that is encoded on it. So I do want you to read that over. Like I said, this is the biology part of DNA. Okay. On the structure. The thing to remember with DNA too, it is a double helix. So double helix means it's twisted. There's two sides to it, and it's twisted. So that's why they call it a double helix. Or sometimes they refer to it as a spiral staircase, because it kind of looks like that. So they call the sides of, they also refer to it as a ladder. So they call the sides of it the backbone, and the base pairs in between, in the middle, that actually code for the different types of proteins that have to be made. Those are the rungs of the ladder. Okay, so it's made up of three things: a sugar and a phosphate, and those are going to be on the side. So if you look up here, This is a phosphate, and that's a sugar molecule, and they make up the sides, and there's one on the other side. And then the things in the middle are the nitrogen bases, and there's 4 different kinds of nitrogen bases on a DNA molecule. If you remember studying DNA in biology, you know that there's also RNA, and RNA is a little bit different, but basically it also has nitrogen bases. It also has a phosphate and sugar backbone, except the sugar is ribose and instead of deoxyribose. That's the difference between DNA and RNA. All right, we're gonna watch this little clip.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
That's really simplified, but this is what they're looking at when they do all the microscope examinations of DNA. So again, there is a Human Genome Project, and if you look at it like it's a file case, this is if you think of a huge cafeteria full of files, that's what the Human Genome Project is. There's enough information to fill up a whole large room in the Human Genome Project files. So again, read this page over just for background on the biological structure of DNA. Alrighty. Now we want to look at it is how is DNA, our focus is always how is DNA used as evidence. So we're going to come back to this in a minute, and I'm going to let our trendy science teacher, is that going to switch? It did not. Yes, it did. Okay, cool. My start button is always behind this little cursor. Oh, I could move it. Hmm. There it is. This is so tricky trying to get that little green button out of my way. It says drag to move it, but when I do that, it comes right back. Ah,
portion of the unit. So we're going to do just a brief overview of DNA. Now, if you remember this stuff from biology, then great. If not, if it's been a while, you may need this refresher. But I feel like it's important for us to first go back and talk about some of the basics with regards to DNA before we jump into how DNA is used in forensics. Ultimately, though, we are going to talk about how DNA is used in forensics, how DNA can be collected from a crime scene, how it can be helpful, specifically how it's extracted from evidence and suspects, and then ultimately how it's linked back to a particular suspect. Now, if it has been a while since your last biology course, or you've never taken biology, then I want you to pause the video and go to YouTube and search Amoeba Sisters DNA. There are some great videos over DNA from Amoeba Sisters, and if you need just a refresher over DNA, definitely check those videos out and then come back to this one for the lesson. All right, so if it has been a while since you have had biology, you might remember that except for identical twins, no two people have the same DNA. There have been many advances in the last 20 years in DNA technology and its use in solving criminal cases. If you join me for the Innocence Project lesson, you know that it's also been instrumental in helping to solve or re-examine post-conviction cases. So DNA is considered individual evidence because it does link to a specific person. Now, all the stars in the sky have to line up for that to happen, but it is possible. So we say that DNA is considered individual evidence. So if I'm a crime scene investigator, I am looking for DNA evidence because it is a crucial part of an investigation. It can really help solidify a case. And so I'm working the crime scene. I'm going to be thinking, where could there have been skin or blood or saliva or urine or semen or hair left behind? And then I'm going to search for that, try to find it so that I can secure that DNA evidence. Now we have something called DNA fingerprinting. You'll sometimes hear it referred to as DNA profiling. It's used in criminal and legal cases to determine identity, or it can be used in cases to figure out parentage. So if you watch the show Maury, you know that it's been used a ton to determine who may or may not be the father of a child. It's also used in some health applications. We'll talk about that in a future lesson, and definitely in crime scene investigation to determine identity or to get a match between evidence and a suspect. All right, so going all the way back to biology where you talked about the cell, DNA stands for deoxyribonucleic acid. It is located in the nucleus of the cell, and we know that it carries an organism's genetic information. The DNA is a polymer that is made up of many monomers. We call these nucleotides. And the structure of DNA can get pretty complicated, but we're going to keep it basic for forensics. So each nucleotide has 3 parts. We have a phosphate group, we have a sugar, which in this particular case we call deoxyribo, and then a nitrogen-containing base or nitrogenous base. Now, one molecule of human DNA contains billions of nucleotides, billions. But there's only 4 types within the DNA. So we have thymine, cytosine, adenine, and guanine. And we use these letters to represent these words because we don't want to write them out every time. And you may remember from biology, A always pairs with T, and C always pairs with G. So it is true that 99% of our DNA is the same from person to person, but there are segments within our DNA. We call these segments short tandem repeats or STRs, which are basically letters of DNA that repeat over and over and over and over. But these STRs differ from person to person. So basically you can take a DNA profile or a DNA fingerprint, which focuses on these STRs.
Pause it there because that's a vocabulary word that you're going to need to put your finger on. So the STRs, remember that word.
That are separated from the rest of our DNA that's the same from person to person, and we can create what's called a genetic profile. If I just confused you no worries. We're going to follow up with more detail in a future lesson. I just wanted to point that out. So the same 4 bases are found in the DNA of all organisms, but their proportions are different. So the sequence of my genetic code is different from the sequence of your genetic code. So in the DNA of each organism, we have adenine and thymine and cytosine and guanine, and they equal according to base pairing rules. And again, you may want to write this down. Apple in a tree, car in a garage. That's how you can remember the base pairing rules. All right, so on a quiz or a test or a worksheet, I might ask you how these nucleotides would pair in a DNA sequence. So I give you one part of the equation you have to complete. So you'll have to use your base pairing rules to answer this. So if you will, pause the video and just write down how these nucleotides would pair.
I'm not going to do that. Basically, what she
All
wants
right, so
you
I
to
want to
do
go
is look
over
at-
the answers quickly so that you can make sure that you know this. So for this first sequence, the nucleotide pair would be TAGCCG ATGCA. For the second one, you should have ATT CGG CTATA.
All right, I'm gonna pause that. I thought she would write it up underneath, but she didn't. So what she's doing is she's taking this whole row, and underneath it, she's saying if I have an A, then that's going to match with a T. If I have a T, it's going to match with an A. If I have a C, it's going to match with a G. And then she's going through the whole row like that. And so she's already on the second row. So I'll follow that with her when she gets to the third one. That as far as base pairing is concerned, it's a rule that A always matches with T. And like she said, if you remember that apple's in the tree, and G always matches with C. So this G is going to have a C, that C is going to have a G, A is going to have a T, T is going to have an A, and so on.
And then for the last one, you should have C, G, T, A, C, G, T, A, T, G. Again, if you're confused by that, go all the way back to the beginning of this lesson and check out those Amoeba Sisters videos because they will help you understand how to pair these. All right, so if DNA is found at a crime scene, we know it belongs to someone, and a match can be found through several steps. So it starts by removing the DNA from the crime scene, and then there's a complicated process that it goes through to match it to a suspect or to a victim. And we call that DNA electrophoresis or gel electrophoresis. And we're going to talk about that in a future lesson. So in comparison to time, DNA is a fairly new technology. So it was developed by Sir Alec Jeffreys and really came into play in the world of forensics in the early 90s. And we've been using it ever since. DNA to this point is considered the most useful form of evidence in obtaining convictions. So it is definitely something that crime scene investigators want to hunt for when they're working a crime scene and trying to collect evidence from a crime scene. All right, so when DNA is found, it's compared to a digital national database we call CODIS. So CODIS stands for Combined DNA Index System, and it contains DNA profiles that have been contributed by federal, state, and local participating forensics labs. So it's just a national database. It's digital. The FBI sort of houses it, and it's used in a lot of different cases. So sometimes the question comes up, what if the DNA is not registered in the CODIS database? There's no way to compare DNA profiles digitally. Well, then investigators have to collect a suspect sample. So without that suspect sample or without that DNA sample being in CODIS, you're not going to find a match. So I know when we watch shows like CSI. It seems as though all an investigator has to do is just swab for DNA, and magically a computer reads the DNA profile and matches it to people in the world, and their picture comes up and their address comes up on a computer screen, and it's just that easy. It is not just that easy. There's quite a few steps involved. And again, you either have to have a suspect sample to compare your evidence sample DNA to, or the suspect has to be in the CODIS database. So how is DNA collected from a suspect? Well, there's a few different ways. DNA can be collected from their hair, from blood, from saliva, but most commonly it's going to be collected in cheek cells. So we call this a buccal swab, where an investigator or a lab analyst just kind of collects cheek cells from the suspect, and then a DNA profile is created from that.
Okay. All right, so that's the first segment.
All right, welcome back to our second lesson
And
over
we're
DNA profiling.
not going to watch that today. We'll watch that tomorrow. Wednesday. But what I'd like for you to do while I'm doing attendance is to, right now this is in the way on this side. Goodness gracious. All right, so I want you to come back to Canvas. I want you to read over as while I'm doing attendance. I want you to read over this segment, and we're going to be looking at, notice she called it fingerprinting. And we're going to do fingerprinting separate, but DNA fingerprinting is a little bit different. So we're going to look at what it means to have a CODIS profile. So read those two segments for me, please, while I do the attendance. So go. And like I said, it was called fingerprinting, but we're not doing it like fingerprint. And when we do, that's our next unit. And when we do that, I'm going to have you do your fingerprints. I'm going to show you how to do that. All right. So again, read that over while I do attendance, and I'll be right back. Kylie, I don't know what you mean by that. Did you miss the quiz? Yes, you did. Okay. You can. All right. So Kylie, as soon as I finish attendance, and I actually have a couple of people who missed the quiz. We took the quiz before the break, and I do have a couple of you. One, Kylie, Lily, are you here today? Nope, she's not. William, you are here today. And Serenity. All right, so I do have 3 of you who missed the quiz before the break. So as soon as I finish attendance, I have to open that up, and I'll let you work on that now. But give me a minute to get to that. All right, yeah, y'all need to jiggle your mouth. I know you were watching the video and you weren't doing anything, but you should be reading now. So I shouldn't be seeing where all these folks are idle. Everybody should be reading. Everybody should be reading. Please look at the Canvas page and be reading. Okay. All right, Serenity, I'll look at that too as soon as I get the test open for you. Okay. Two. Missing somebody. Can't tell who it is right now. Hmm. Okay, I'm just gonna leave it like that. It'll be fine. Okay. Why does it do that to me every time? Actually, it should already be open. So I just need to get, because it's, according to this, it's open until Friday. So you just need a code. All right. So this is William Serenity, and I'm putting the code in for everybody, but this is for those of you who need to take the test. There it is. All right. Should be 3 of you taking the test. William, Serenity, and Kylie. The code is in the chat. It should be open. Let me know if you can't get in it. According to this, though, it should be open until Friday. And it shall say serology test. Serology, what does it say? Forensic serology is the name of the test. All right, William, I hope you're there. You're not just logged in. All right, Kylie's in. Serenity's in. So, William, it's just you. I'm just waiting on you. Okay. Let's see what time is it. Okay, everybody else, please read those 2 sections. All right, I'm going to, hopefully you were reading. All right, since we have some folks who are testing, I'm just going to share this one with you. It's taking forever to share.
You've probably heard of the human genome, the huge collection of genes inside each and every one of your cells. You probably also know that we've sequenced the human genome. But what does that actually mean? How do you sequence someone's genome? Let's back up a bit. What is a genome? Well, a genome is all the genes, plus some extra, that make up an organism. Genes are made up of DNA, and DNA is made up of long paired strands of A's, T's, C's, and G's. Your genome is the code that your cells use to know how to behave. Cells interacting together make tissues. Tissues cooperating with each other make organs. Organs cooperating with each other make an organism. You. So, you are who you are in large part because of your genome. The first human genome was sequenced 10 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries, and cost over $3 billion. But someday, very soon, it will be possible to know the sequence of letters that make up your own personal genome all in a matter of minutes, and for less than the cost of a pretty nice birthday present. How is that possible? Let's take a closer look. Knowing the sequence of the billions of letters that make up your genome is the goal of genome sequencing. A genome is both really, really big and very, very small. The individual letters of DNA, the A's, T's, G's, and C's, are only 8 or 10 atoms wide, and they're all packed together into a clump, like a ball of yarn. So, to get all that information out of that tiny space, scientists first have to break the long string of DNA down into smaller pieces. Each of these pieces is then separated in space and sequenced individually. But how? It's helpful to remember that DNA binds to other DNA if the sequences are the exact opposite of each other. A's bind to T's, and T's bind to A's. G's bind to C's, and C's to G's. If the ATGC sequence of 2 pieces of DNA are exact opposites, they stick together. Because the genome pieces are so very small, we need some way to increase the signal we can detect from each of the individual letters. In the most common method, scientists use enzymes to make thousands of copies of each genome piece. So we now have thousands of replicas of each of the genome pieces, all with the same sequence of A's, T's, G's, and C's. But we have to read them all somehow. To do this, we need to make a batch of special letters, each with a distinct color. A mixture of these special colored letters and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome. This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book written in a language you don't speak. You can recognize all the letters, but still have no idea what's going on. So the next step is to decipher what the sequence means, how your genome and my genome are different. Interpreting the genes of the genome is the part scientists are still working on. While not every difference is consequential, the sum of these differences is responsible for differences in how we look, what we like, how we act, and even how likely we are to get sick or respond to specific medicines. Better understanding of how disparities between our genomes account for these differences is sure to change the way we think, not only about how doctors treat their patients, but also how we treat each other.
Okay. All right, so that's just a little info on what the human genome, because they're going to talk about that a lot. One of the things she talked about also um, was electrophoresis. And so we're going to actually look at an activity. What time is it? Where you'll see what electrophoresis is.
In this video, we're learning the parts of the cell. Now remember,
All right. So again, I want you to focus on this part right here for today. What exactly, as far as forensic is concerned, where are we getting the DNA information from? And then we're going to look at how we're going to analyze it. So if there's a weapon, we can look at the weapon for skin that might have been left behind, blood stains, sweat, and any other body tissues that would be. Clothing, hat, mask, they also contain hair samples and sweat. Again, your sweat has cell droplets in it. Sweat is a waste product from your body, so it has a lot of cells in it that will have your genetic information in it. Bathroom supplies such as tissue, cotton swabs contain bodily fluids. It's one of the things you see in those crime movies is somebody was drinking a soda and they wanted to get a sample from them. They happen to put it in the garbage. They'll take the bottle out of the garbage, hoping that the saliva from their mouth is left on the bottle or the cup or whatever. Um, Things like mucus and airwax or blood is probably something you would get from the body. And these types of things, like they said, would be compared to a CODIS database. Now, like I said, for crimes that they, I was gonna say cold case crimes, that where they had samples from the victim stored up, and then they all of a sudden have somebody else show up, another that they just locked up the other day, and they take samples from them and they compare their samples, and all of a sudden these samples match the samples we had from 20 years ago. And so that's how they are able to match those up. Toothpicks, if you ate cigarettes, um, contraceptives, the linen on the bed, what you leave behind in the bed, fingerprints, um, obviously, may also, fingernails, like if the person scratched the other one, you'll have cells underneath their fingernails. So all of these are places where you can find DNA evidence on the victim or on the suspect. And what they were saying too is sometimes what they do is if they suspect a person but cannot get a sample from that person, they can call in family members, a brother or sister or someone like that, and get a sample from them, and the match will be similar, if not exact. I mean, it won't be exact, but it will be similar enough to make a connection. Right. So again, it's um, it's like we did blood typing. This is a similar thing, whereas you want to uh, line it up in such a way that you can read the sequence. So this picture over here is a map. I don't know if you remember from genetics, you might have done karyotypes where they take all of the chromosomes and line them up and number them. And so you knew which one. So this would be like a karyotype. And the position of the genes on each chromosome is unique. And then you have your X and Y chromosomes at the end here that tell you whether it's a male or a female. And so there, and then they mentioned in the video the STRs. Okay. So I told you to remember that short random repeats. So this is what they're looking for when they're trying to identify. Because as they said, there's only four different bases in DNA, but it's like how all of you have, there's no two people in this class that have the same name, but we only use 26 letters in the alphabet to spell a word. Okay, so that's the same concept with DNA. There's four letters, but depending on how you arrange them, the sequence that you arrange them in, will give you different information. Okay, so they look for the short tandem repeats, and that's an identifying factor. Okay, because like you said, we're human, so for the most part, the sequence to make an eye, the sequence to make an arm, the sequence to make a nose is similar. There might be certain uniqueness about the instructions on yours that tells you exactly how your nose is made, but most of that DNA is going to be common to everybody because we all have an eye, a nose, an ear. Um, we all have saliva. So it's all going to be similar, but there'll be certain unique sequences that will identify you. All right. And again, because people are testing, I don't want to get too far ahead. So I want you to go through and um, do this review activity, read this profile, and then we're going to talk about um, electrophoresis next time and how they actually uh, spread it out and read the DNA on the lab test. All right, so for today, just read up, and you can go as far forward as you want. But just do some reading. The vocabulary, like I said, the vocabulary for this unit is a lot. So spend some time with the vocabulary, if nothing else. Don't worry so much about the structure because, but do, all right, so for you all, start from where number 11, go down, okay? As far as vocabulary, start from number 11, go down. Everything else really just has to do with genes and stuff up here. So this is the biology part of it up here, but this is the forensics part down here. So with the time that's left, go ahead and look at that. Like I said, I'm not trying to get too far ahead because we got a couple of people testing. Oops. Stop sharing. There we go. Mm. All right. Sorry, Lorenzo. I didn't see you there. Lorenzo, we're on DNA analysis module. If you would read through the first 3 pages of that, please. All right, and I need to check. Some of y'all submitted some things. All right, the other thing, the study guide really had no point value to it, but if you bothered to submit it, I did give a point for it, so that's a bonus point actually. So no harm, no foul if you didn't submit it, but you did get a point if you did. I gave more than a point. Like some of you actually have an A, so it was taking you way over 100. The grade book doesn't like that when you go over 100, but it's there. But that will balance out when you get another grade that isn't 100. So I'm leaving it there. So again, read over those sections. We're going to look at next class. We're going to look at, oops, I'm sorry. I thought I turned my phone off. Apologize for that. Those pesky spam people. Anyway. What was I going to say? So we're going to look at some lab things on Wednesday when you come, how they actually do testing for DNA. So again, just spend some time with the vocabulary and that's where we'll pick up on Wednesday. All right, so if you have no questions for me, those of you who submitted things, I am checking on that now. I'll get that caught up in a second. But if you're good, I'm good. You all can go ahead and sign out and have a great rest of your day.